Good morning, everyone. My name is Ying. So I'm a PhD student from the University of Queensland, IMB, and today I'm going to talk about a part of my PhD project titled Methylation Profile Scoring Model for Discovering Epigenetic Evidence on Endometriosis. Basically, I'm working on a disease called endometriosis. So endometriosis is a type of gynecological disease in which the lining of the uterus can be found outside of the uterus and form lesions. It is estimated that around 5 to 10% of reproductive age women worldwide are affected by this disease. And currently we know that endometriosis has 3 common lesion types, namely SUP, OMA, and DIE. Recently, there's a new theory on the pathogenesis of endometriosis have been proposed called the genetic epigenetic theory. So what this theory says is that endometriosis starts with endometrium cells or the precursor of endometrium cells that have inherited genetic and epigenetic defects. So this defects increases the chance for these cells to transform into a microscopic lesions. And how endometriosis starts is with the presence of these additional genetic or epigenetic changes on these microscopic lesions that transform them into more severe lesions, which eventually leads to endometriosis. So the main focus of my project is to identify epigenetic changes or defects that are related to endometriosis. Specifically, I'm looking at a type of epigenetic called DNA methylation. So as we all know that DNA methylation can change gene expression, and a change in gene expression could potentially be a cause of the disease. And so the motivation of my project is to identify specific DNA methylation sites that are associated with endometriosis. And the classic way of doing this is to perform an MWAS, methylome-wide association studies. But the problem with this method is that a large endometrium tissue sample size is needed. And currently we don't have this much sample size. And so what I'm trying to do is to see if we could use other methods, a more highly powered method, to see if we can detect DNA methylation sites that are associated with endometriosis without the need of increasing sample size. And the method that I'm using here is the methylation profile score method. So methylation profile score is a value that can be calculated for each individual. And this value represents the particular individual's DNA methylation patterns. So what I'm trying to do is to develop a methylation profile score for endometriosis. What I mean by this is I'm trying to calculate a score that represents an individual's risk of getting endometriosis based on their DNA methylation pattern. So how can I use this method to help me identify specific DNA methylation sites that are associated with endometriosis? Is first of all, in order to calculate methylation profile score, we need to know what are the methylation sites that we want to include in calculating the score. And theoretically, a good performing methylation profile score for a particular disease derive from methylation sites that are associated with the disease. So what I did is I calculate different versions of methylation profile score using a combination of different methylation sites. I evaluate their performance, and then I pick those scores that have the highest performance, trace back, and see what are the methylation sites that I use to calculate the score. And from there, we can know what are the methylation sites that are associated with the disease. So in my project, I analyzed 2 different data sets separately. So the first data set is the DNA methylation data derived from endometrium tissue samples. So we have a total of 951 samples. All of these samples come from 4 different cohorts, namely Edinburgh, Melbourne, Oxford, San Francisco. And on the other hand, we have around 291 blood tissue samples, DNA methylation data analyzed. However, for our blood tissue samples, all of them only come from a single cohort, which is the Melbourne cohort. So a little information about the datasets I use. This is the mean age for endometrium tissue samples, which is 35. Blood samples, 30. Exclusion criteria are hormonal treatments, irregular menstrual cycles, signs of cancer. So we've analyzed the DNA methylation data of our data sets using Illumina microarray. And after QC, we have a total of around 700,000 DNA methylation probes to analyze. And apart from collecting DNA methylation data, we also perform genotyping on our samples as well. So these are QC parameters that we use. So after QC, we ended up with around 6 million variants. After we QC our DNA methylation data, we remove batch effects using SVA analysis. And before we proceed with calculating the score, we just want to make sure that DNA methylation from our samples can capture variants in endometriosis. And we estimate this by using the ORAMO model from the OSCAR software. So this is the results. Y-axis represents the estimated proportion of variants in endometriosis that can be captured by DNA methylation. The x-axis shows the different models that I use to estimate proportion. Red color represents blood tissue samples. Green color represents endometrium tissue samples. So overall, what can we see is that in the more complex model, after remove batch effects, after correcting for population genetic structure, and after correcting for age, we identified that around 13% of these variants in endometriosis can be captured by DNA methylation from endometrium tissue samples, which gives us a green light in calculating the score derived from endometrium tissue samples. But the problem is, as you can see, that currently we just don't have enough power to estimate the proportion of variants in endometriosis captured by DNA methylation from the blood tissue samples. And so the question here is that why and now result in the blood tissue samples? Is it because of power issue, or is it because DNA methylation from blood just couldn't capture any variants? And so in order to do this, we perform the same analysis, this time using endometrium tissue samples with a smaller sample size and see whether the results is similar. So the blue results here shows endometrium tissue samples with a smaller sample size. So although if you look at the model over here, endometrium tissue samples with a smaller sample size estimated around 20% of proportion of variants in endometriosis captured. If you look at the number itself, there's a really huge difference between them. However, if you look at the error bar, a huge error bar actually suggests that there's not much difference statistically. And so what this means is that potentially a null result shown in blood tissue samples could be because currently we just don't have enough sample size to estimate the proportion of variants captured. So this slide shows the specific steps that I used to calculate the methylation profile score. So in order to calculate the score, I divide my samples into two groups. One's a test group, one's a training group. The purpose of a training group is to estimate the effect size for each DNA methylation, or we call it as weight, for each DNA methylation. And this information will be used in calculating the methylation profile score on individuals from the test dataset. So the blue box here shows a list of different versions of methylation profile score that I calculated. All of them differ in terms of two things. First is the method that I use to calculate effect size. So the model that I use to calculate the effect size is the MOA and the moment model. And the second thing is that the p-value threshold. So what p-value threshold here means is that this is a threshold that I use to identify what methylation sites that I want to include in calculating the score. So ideally, in order to properly evaluate my methylation profile score, we need our tests and training data should be coming from an independent cohort. And so what I did for endometrium tissue samples is I split my samples into tests and training based on which cohort those samples came from. And so what I did is samples coming from Edinburgh, Oxford, San Francisco are all categorized under the training dataset. And then samples coming from Melbourne are categorized under the test dataset. However, as all our samples from blood tissue samples coming from only a single cohort, so what I did is I randomly separate them into half. Half of them used as a test dataset, another half used as a training dataset. And this is my results. So y-axis here shows the AUC. So this represents the performance of the methylation profile score in predicting an individual's endometriosis status. Of course, the higher AUC, the better. And the model that I use to predict an individual's endometriosis status is a logistic regression model. So what this model here says is that I'm using an individual's methylation profile score plus their age to predict their endometriosis status. So if you look at our highest performing methylation profile score has a AUC value of 0.5841, which I would say is fairly good because based on previous studies on methylation profile score on complex diseases, their AUC usually range around 0.6. And if you look at the p-value, so what is p-value here means is that it's a p-value for the logistic regression model. So a p-value of less than 0.05 means that my score is significantly associated with endometriosis. But since we're using this method to detect specific DNA methylation sites that are associated with endometriosis, so our p-value threshold should be corrected for multiple testing. And obviously, our p-value doesn't cross the multiple testing threshold. But then remember, all these results derive from a low sample size with less than 1,000 samples, which is really low for an analysis like this. And given such a low sample size, we can still detect minor signals like a bell-shaped curve pattern as we increase the methylation size we use to calculate the score. And the lower bound of our 95% confidence interval for most of our AUC are mostly more than 0.5. These minor signals suggest that we could still potentially use methylation profile score derived from endometrium tissue samples to help us detect specific DNA methylation sites that are associated with endometriosis. It's just that currently we might need to tweak our model here a little bit to increase prediction accuracy for the model to be valid enough for us to determine specific DNA methylation sites that are associated with endometriosis. And this is the results performance of my methylation profile score derived from blood tissue samples. And so obviously, although we have a fairly good number of AUC and a p-value less than 0.05, but if you look at the lower bound of our 95% confidence interval for most of our AUC over here, most of them less than 0.5. So what this means is that all of our models over here is not performing really well. But the minor signals collected here and there could suggest that DNA methylation derived from blood tissue samples could still potentially capture some variance in endometriosis. It's just that currently we just don't have enough sample size and power to use this method to detect sites that are associated with endometriosis. So the limitations of my study is that DNA methylation fluctuates throughout an individual's lifetime. So it could be both the cause and a consequence of the disease. And also we should adjust for cell type, but we don't have a cell type proportion reference data. And in summary, although we currently don't have enough power to develop methylation profile score for blood tissue samples, but our methylation profile score derived from endometrium tissue samples performing fairly well. And so moving on, we are planning to do is to tweak the model a little bit to increase prediction accuracy so that we can have a valid model to detect specific DNA methylation sites that are associated with endometriosis. So before I end my presentation, I would like to thank my supervisor for their guidance along the way, and also our collaborators for collecting and sharing the data, and last but not least, friends from IMB for the emotional support. Thank you.
Thank you, Li. We have time for one or two quick questions.
A great talk, by the way. Just quick question. Is there any relationship with DNA methylation and SNP in endometriosis? SNP, single nucleotide polymorphism. Is there any association? Did you see it?
Endometriosis and SNP.
So association with DNA methylation and SNP.
Yes. So we did have a study where we try to determine the association between DNA methylation and SNPs, which we call it as an MQTLs. So it's a study that's been done by our group, and yes, we do. Yes, we do have.
And one more very quick question.
Hi, nice talk. So my question is, when you show this pie chart here, when you show this pie chart where you show like you have samples from different places, I was wondering why did you split the samples from one specific location into test data set? So why don't you take a subset of the samples coming from all the places, or am I missing something here?
Yes. So when you want to have a test and training data set, we should have both of these data sets should be coming from an independent cohort. So it should be come from a totally different population. So for endometrium tissue samples, actually is actually collected by a few of our collaborators. So all of them are collected from a different population. And so and so that's why when I'm splitting test and training, I split them based on their cohort.